Hello guys, Assalamu alaikum. I hope you all are doing great and studying hard. So your Edexcel IAL Biology Unit 2 exam is approaching, and today I'm here with another valuable video, a full syllabus review of Unit 2. It's a detailed topic by topic revision session to make sure you have covered everything important before the exam. So without further ado, let's get started and make the last few days count. So if you start here, we know we have few types of cell. So we have bacterial cell, a bacterial cell basically, animal cell, plant cell. Okay, so we'll be discussing their characteristics, their structure and all. So let's start with the bacterial cell, which is like the most ancient or the earliest cells and the simplest one. So for bacterial cells, they have cell wall, cell membrane, ribosome. Sure. Yeah. So let me start with cell wall. Yeah. So let me start with cell wall. So we know cell wall, it's the same for all plants. It's the same for plants and bacteria, but the thing that the cell wall is made of is different. Okay. So their cell wall is made of peptidoglycan. Okay. We know plant cell wall, it's made of cellulose and all, but for bacteria, their cell wall is made of peptidoglycan and it's made from murin that will prevent the swelling and bursting of bacterial cell. And it will help keep its shape and protection to the cell contents. Okay, so it will protect the cell contents. It will help keep the shape of the cell, the bacterial cell, and it will prevent the swelling and bursting. Okay, the swelling, bursting, all this, the function is the same as plant cell wall, and this one just the constituent of it is different. And then we have cell membrane. So the normal function, as in the one that we know for plant cells and animal cells, that one. Plus, they have respiratory enzymes. Okay, so it's side of respiratory enzymes. And we have ribosomes. So ribosome is 70S. They have 70S ribosomes basically. And we know in ribosomes protein synthesis occurs. So that's the side of protein synthesis. We have mesosome. So the inner foldings that you can see here, this infoldings or inner foldings, this one, basically the cell membrane, it gets inside, it folds inside, and this thing forms the mesosome. And yeah, it's a photosynthetic membrane. And for the DNA, they have loop of DNA. So their DNA is like this. It's a loop of DNA and it has no histone proteins. And whenever it has no histone proteins, we call it naked DNA, okay? So it's not covered with histone proteins. And flagellum, I'm sure you know what a flagellum is. So basically this long extended part from the bacterial cell membrane. This is the flagellum and it beats so that the bacteria can move and swim. Okay? It helps it to swim and move. For pili, yeah, so flagellum and pili, these two might seem similar, but the function is different. Okay, flagellum is this one here, pili is here, this one. So pili, they basically attach to the host cell, okay, or to the surfaces. Okay, it helps the bacteria to attach to the host cell or to any other surface and hold onto that surface. Okay, and the capsule, which is the outer layer here, and it helps them survive the dry conditions. If, for example, in desert areas or any other dry conditions, if the bacteria is in a drier condition, it will help them survive. And it will basically, by preventing dehydration, it will help them survive. And it protects from the phagocytes. So we know the phagocytosis, that thing. So the cells that do phagocytosis is phagocytes. It will help them from phagocytes. And there's this plasmid. Yeah, so there's this plasmid. That's the extra one. And you don't find this in other cells, but plasmid is only in bacterial cells. So they basically carry the genes and they will make the bacteria resistant to antibiotics. Okay, remember, we know about antibiotic resistance and all. So the resistant thing, the structure that does the resistance, that makes the bacteria resistant is the plasmid, okay? And it's needed in protein synthesis. And I think I said, yeah, I talked about all of them. So for peptidoglycan cell wall, bacteria, they have peptidoglycan cell wall, and there are two types, okay? There's gram-positive and there's gram-negative. Okay, so this is basically the difference in the color mostly. So for the gram-negative, sorry, the gram-positive, let's start with the gram-positive first. Yeah, so the gram-positive, they are basically the thicker cell wall, and they have teichoic acid. And yeah, so they have a thicker cell wall, so they can retain the violet color, okay? And that's why if you put it onto a microscope, the cell will appear violet, okay? So that's a gram-positive bacteria. And for the gram-negative one, they have a thinner peptidoglycan cell wall, and they have no teichoic acid, okay? So as they don't have any acid and they have a thinner cell wall, the violet color, it will not be retained. So that's why it will become pink or it will be shown pink in the microscope. Okay, if you see it using a microscope, it will be shown pink. And as I always tell you, these notes, the revision notes, they're very, very important because I did not include everything in the revision notes. Okay, here I would discuss everything from the full syllabus because that would take a lot of days. But here in this revision class, we'll only discuss the important ones. So make sure you know this important ones. Okay, these are like the bare minimum that you should know. So make sure you know them, make sure you memorize them, and I'm doing it earlier this time so that you guys have time to go through it, okay? And then we can, so that you know what's in the syllabus, and then when we're doing questions, you can relate this to the ones that we're doing, okay? The questions. And in the questions, you're gonna see this question comes a lot, like the information that's in these notes, they come a lot. Mostly they come, but of course, there are gonna be questions from outside here. But these are the more important ones, okay? And yeah, so the other two types of cell, animal cell, plant cell. So let's start with animal cell. So this is how an animal cell looks like. So animal cells, they have double membrane bound organelles and single membrane bound organelles and no membrane bound organelles, okay? So if they have double membrane, it's called double membrane bound organelle. If an organelle has one membrane, it is called single membrane bound organelle. And if it has no membrane, it's not enclosed by any membrane, that's called no membrane bound organelle. Okay, so the blue ones, these are the double membrane bound organelle. And the pink ones, they're the single. And the green ones, they're the no membrane ones. Yeah. So let's start with the single. Actually, no, let's start with the double membrane bound organelles. So we have mitochondria, we have nucleus, and animal cells don't have chloroplasts. So yeah. Okay. So for animal cells, it's only mitochondria and nucleus. Okay, a mitochondria and nucleus. So let's start with the nucleus. So there's one here, it's the nucleus, and here it's the nuclear envelope, the covering of nucleus, it's the nuclear envelope, and in between the covering, there's spaces or pores, which we call as nuclear pores. And the nuclear envelope, it contains chromatin. Here inside it, it's the chromatin. And the darker one, usually if you see it in a microscope, the pink color area, it's the darkest one in the nucleus. So that darker part is called the nucleolus. And the nuclear pore, one more thing I forgot to mention, the function is that it will allow the RNA to leave the nucleus. Okay, after RNA is made here, DNA replication and all, for transcription and translation, it has to go out, right? So that's why the RNA, sorry, after transcription, it has to go out, right? So the RNA, after it's been made in the transcription part, it will leave and go to the ribosomes, okay? And it will go to the cytoplasm where there's a ribosome and all, so yeah. And for what else we have for mitochondria, here we have a structure of mitochondria. So mitochondria has a outer membrane and an inner, actually it's a double, it's a double membrane. So it's gonna have two membranes also. It's gonna have an outer membrane and an inner membrane. So the outer membrane is the plain surface that you can see, but the inner membrane, it's this one here. And Yeah, and the infoldings here, the wavy structure here in the inner membrane, it's called crista or cristae. Okay, and it has 70S ribosome, like bacteria. So, can anyone tell me what other organelles or what has 70th ribosome? Can you guys give it in the chat box? Or you can see if you want. What other organelles or structures chloroplast? Yes, very good. And do anyone else have the same answer, different answer? Yes, prokaryotic cells. Anything else? Anyone? Acha. So basically, yes, the three things: mitochondria, chloroplast, they have 70X, and the prokaryotic cell. So bacteria was an example of prokaryotic cell. So bacteria also has 70S ribosomes. Okay. So these three, they have 70S ribosomes, and other than that, all the eukaryotic cells, they have 80S ribosomes. Okay. Even though mitochondria and chloroplast, they're part of the eukaryotic cell, which is animal cell, plant cell, but still they have 70S ribosome, because this is theory that we know. So further theory that they evolved from prokaryotic cells. That's why they have similar characteristic to prokaryotic cell, which is 70S ribosome. And yeah. And yeah, so the crystalline foldings, matrix. Okay, so the cytoplasm or the jelly-like liquid in the mitochondria, that's called matrix. And the jelly-like liquid in each of the organelles, it has different names. So for mitochondria, it's matrix. For cell, it's cytoplasm. And mitochondria, it has a loop of DNA. And we know the function of mitochondria, the main function is that it synthesizes ATP by aerobic respiration. And mitochondria, it has a rod shape. And yeah, that's all. I should describe the site of, so mitochondria, we know it's a site of aerobic respiration. It synthesizes ATP, and it has a role in the lipid synthesis also. Okay, so it synthesizes lipid, it synthesizes ATP, and it's a site of aerobic respiration. So that's it for mitochondria. So that's the two double membrane bound organelles. For single membrane bound organelles, we have smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi body, lysosome. Okay. So let's know about smooth and rough. So there's like there's endoplasmic reticulum, and it's divided into two types: smooth and rough. Okay, if it has ribosomes on it, it's rough. If it does not have a ribosome, it's smooth. Okay, so the rough one with ribosome, it's flattened and it is interconnected sacs. It has flattened interconnected sacs, which are called cisterna. Okay, don't confuse crista and cisterna. Okay, crista is for mitochondria, crista or cristae, and cisternae or cisterna. This is for rough endoplasmic reticulum. Okay, it's the inner foldings of that. Sorry, not the inner foldings, the sacs basically. They're called cisterna or cisternae. And for smooth endoplasmic reticulum, they won't have ribosome, and their function will be to make lipids and steroids. Okay, so that's their function. And then Golgi body and lysosome. So Golgi body and lysosome. Actually, lysosome, it's basically a vesicle, and it will contain digestive enzymes. Okay, the vesicle will contain digestive enzymes. And for Golgi body, we know the Golgi body, after a protein is made, it's sent here, and it will modify the protein. It will package it into vesicle, and yeah, then it will transport it to other parts. And that is Golgi body. It's also made of flattened curved sacs, but they're parallel. Okay, they're parallel, and they have different sizes with a smooth single membrane. So yeah, for rough and like endoplasmic reticulum and Golgi body, one of the very big difference is that for endoplasmic reticulum, you're gonna see the sacs, they're connected, okay? And for Golgi body, you can see they're different individual sacs. Okay, so that's the difference between Golgi body and endoplasmic reticulum. And beside Golgi body, you're going to see vesicles always. But for endoplasmic reticulum, you're not going to see vesicles. So here, this is the structure of Golgi body or Golgi apparatus. It has vesicles and cisterna. Okay, yeah, I've discussed everything else. You don't need to know more. So that's it for the single membrane bound organelles. Let's go to non-membrane bound. So ribosomes, we know translation occurs in ribosome. And nucleolus, the one they're saying it's inside the nucleus or nuclear envelope, and that will make the ribosomes, okay? And then ribosomes, they do the translation. So yeah, both the nucleolus and ribosome, they don't have any membrane. And nucleolus, it's a site of transcription, okay? So it will have 9 triplets of microtubules, and they'll be arranged 90 degrees, okay, at right angles. Okay, so this, if I say this is a microtubule and the other one will be arranged here, okay? So 2 microtubules arranged at 90 degrees to each other. That's for centriole. So centriole will be made of 9 triplets of microtubules. So remember that this diagram that I drew here, this will have microtubules like this. So there will be 9 triplets. Okay, 9 times 3. So there will be 9. 1, 2, 3, 2, 2, 3, 3, 2, 3, 4, 2, 3, 5, 2, 3, 6, 2, 3, 7, 2, 3. Actually, I don't have space, but like this, there will be 3 into 9 microtubules. And the centrioles, as I say, they'll be arranged 90 degrees. So this is one centriole, this is one centriole. So they'll be arranged 90 degrees. Okay. And they make their function is that they will make the spindle fibers that attach to the centromere. And we know during cell division, the centromeres, they separate the chromosomes. Like the chromosomes are separated at the centromere that's attached by the spindle fibers. They will pull it to opposite poles and all. So that spindle fiber is made from centrioles, okay? And rough endoplasmic, which I've missed this part for rough endoplasmic reticulum, that they have ATS ribosome because it's a eukaryotic structure. That's why ATS. And it will fold into 3D or 2D shape, and it will package to Golgi, okay? So yeah. And okay, and also it stores and transports protein, okay? That's like an extra information, but yeah. Stores and transports protein. I think I said everything for plants. Oh, sorry, animal cell. Yeah. And then for plant cell, let's go to the last type of cell, which is the plant cell, which you're very familiar with. The plant cells, we already discussed a lot of them. Golgi vesicles, ribosomes, smooth endoplasmic nucleolus, nucleus, rough. Okay, so one difference is that they will have a large central vacuole. Okay, for plant cells, do they even have vacuole? Nah, in this one, there's no vacuole. But mostly for plant cells, they have large central vacuoles. For animal cells, they either have no no vacuoles, or even if they have it, that's very small, okay? Very, very small, which is negligible. And yeah. But for plant cells, they have very large vacuole, like this part that you can see here, the whole thing. This is the vacuole, okay? So it's very large and it's in the center. So that's where a large central sap vacuole, and it has a myeloplast. This is also important. A myeloplast question came before also a lot of times. That a myeloplast, they will contain the starch, okay? A myeloplast contains starch. Cytoplasm, mitochondria, same thing, graphite crystal, vacuole membrane. Chloroplast is another extra structure here. So chloroplast, it's only present in plant cells because we know chloroplast, it's a site of photosynthesis, and only plants, they can photosynthesize. So that's why chloroplast will be present in plant cells. Golgi apparatus, okay, everything same. Also, the membrane for vacuole, we know the name is tonoplast, okay? The vacuolar membrane, it's tonoplast, and we need it for turgidity, okay? It gives the vacuolar cell turgidity, and it will store water, sugar, salts inside. Yes, it's a storage again for water, sugar, and salts. And this basically lowers the osmotic balance. Okay. And if it's basically if it was high water inside, the water would have gotten out by osmosis or like moved out by osmosis from the higher water potential to the lower water potential down the concentration gradient, and the plant would have shrinked, okay, or become plasmolized. Yeah. The plant cell would have become plasmolized. So in order for the plant cell to become plasmolized or dehydrated and all, they have a tonoplast, and that basically stores, or more like the vacuole stores water, sugar, and salts. And that basically increases the salt or sugar concentration and lowers the water concentration so that there's a balance, okay? So that the plant cells, even if it has less sugar inside and more water outside, even if water comes inside, as it has a cell wall, the cell wall will prevent the plant cell from bursting. Okay, so that's why the plant cell does not burst. Okay, even if water has entered. So that's why, and that's what is held by the vacuole and the chloroplast technique. Okay. And what is this one? Oh, sure, chloroplast structure. So let's get into the depth of the chloroplast structure. So chloroplast, we already discussed that it's a double membrane bound organelle. So it will have two membranes. We can see easily it's visible that two membrane. So the outer membrane, inner membrane. Actually, for mitochondria, the outer membrane was smooth, but the inner membrane, it was folded inwards, right? But for chloroplast, we can see that both the membrane, they're smooth, okay? None of them are infolded or all, so both are smooth. And it will also have sabotage ribosomes. And what do we have? Acha. So thylakoids. So thylakoids are these structures, this sac-like thingy. And all these thylakoids together, they form granum, okay? This is one is called granum. Lot of them together is called grana, okay? And the outer membrane, inner membrane, and the space between the membrane, this part, it's called the intermembrane space. And the inner membrane stroma is the jelly-like liquid or the fluid of chloroplast, okay? For mitochondria, it was matrix here. For mitochondria, it was matrix. But for chloroplast, this is stroma, okay? And there's a loop of DNA, there's intergranal thylakoid. Yeah, and the thylakoid, it will basically store the chlorophyll pigment, and we know chlorophyll, it traps the light energy, and that will be used for photosynthesis, okay? So that's the function of thylakoid. And yeah, so do any of you have any questions for the three types of cells? If not, then I'll move on to the next one. Okay, so comparison between microscope. So we know microscopes, there are two types of microscope, light microscope, electron microscope, and between or within electron microscope, there's again two types, scanning and transmission. Okay, scanning electron microscope, SEM, and transmission electron microscope, TEM. Okay, so with the medium is different. So for light microscope, it's light. For electron microscope, it's electron. Okay, from here, that's how we get the name for electron microscope and light microscope, because the medium is different. And for dimensions, we will see 2D pictures from using light. Running out of time. Yeah, we will see 2D pictures of from light microscope. But for electron microscope, we can see the 3D. For scanning, we can see the 3D, but for transmission, we can again see the 2D structure, okay? So only the scanning electron microscope, it gives us a 3D structure. And the magnification, so that's the maximum magnification. So for light, it's times 1500 to 2000. For scanning, it's 200,000. And for transmission, it's okay. So for resolution, so for light microscope, it's 200 nanometers, scanning 20 nanometers, transmission 0.1 nanometer. And specimen, so it's the specimen is what we're viewing under the microscope. So for light microscope, we can either see the living specimen or the dead specimen. But for electron microscope, we can only see the dead specimen because when electron goes to the electron microscope, electronic microscope or electron microscope, that time in vacuum and all, it needs to be done in vacuum, and the cell, it will basically die. Okay, the cell cannot leave. So that's why we can only view the dead cell using electron microscope, but we can see the living and dead both for light microscope because light does not kill. And here we are given the images. So the image that we see from microscope, it's called micrograph. So this is how we would see for light and electron and transmission. So for scanning one, one more thing that you can see is that we can see the surface of a structure, okay? For scanning electron microscope, we can see the surface of a structure, but for transmission, we can see the inner structure, okay? Not the surface, but the inner structure, okay? That's one more difference. And here we have the cells. So electron microscope, yeah, this is an electron micrograph. So animal cells, this is how a general animal cell looks like. A plant cell. This is the Golgi apparatus with vesicles in the side. This is the chloroplast. We can see the grana visible. The mitochondria with the crystal visible. And endoplasmic reticulum, basically the sac-like structures. Yeah, so this is it for this one. So do any of you have any questions from the cell ultrastructure? If you don't, then I will go on to the next note. Okay, moving on to the next one. Gene expression. Okay. So gene expression, transcription of DNA by mRNA. So we know transcription occurs, and after transcription, it's translation. So firstly, it's the replication where DNA will be copied or replicated, and that copied DNA will then go under transcription, and it will form a mRNA. And that mRNA will be translated, and it will be formed into a protein. Okay, so that's the process. And controlling gene expression inside and controlling gene expression outside. Okay, so there's two things, inside and outside. So for inside, the transcription factors, the proteins, it will bind to the specific sequence of DNA base. And yeah, so the RNA, the premature RNA and the RNA splicing will occur, we know, right? Introns, exons, and all that. And for the outside one, there'll be the post-translational change where the polypeptide chain length will be changed. Okay. That's what happens outside. So let's go to the details of inside the nucleus. So transcription factors. So a protein which will control the transcription of a DNA from DNA to mRNA. I told you that's what transcription is, by binding to a specific sequence of DNA bases. So here we have the enhancer. Yeah, enhancer, the promoter region, the gene, introns, exons. And so the transcription factor, it will bind to the promoter region and it will initiate the transcription. Okay, when it binds to the promoter region here, it will initiate the transcription and it will allow the RNA polymerase. It will bind to the RNA polymerase, will bind to the promoter. And the transcription factor, it will either bind to the enhancer and promoter. So if it binds to the enhancer, it will regulate the activity of transcription by allowing the DNA to open chromatin or closed. Sorry, to be an open chromatin or closed, yeah. And for promoter one, we already said that it will initiate the transcription. So enhancer will enhance it. Promoter will start or promote the transcription. Yeah, and for RNA splicing, we know the introns. This part, this is the intron. So it's the unwanted gene or the useless, the non-coding part. So that is unwanted, so it will be removed. Okay, we don't need any unwanted part in our genes. That's why it will be removed. And the exons, they'll be joined together. Okay, and they'll be reattached by spliceosome. So this is enzyme for spliceosome. It will reattach it. And it will remove the unwanted non-coding introns. So the one that's saying it will remove it. And will join the exons together. And the mature RNA, it will then be translated. And it will allow a single gene to make several versions of functional DNA. Okay. So one gene can make many versions of functional DNA. Okay, so this says here the exons, it will be cut or removed. And mainly the introns are cut, but if there's useless exons, they will also be cut or removed. And the exons, they're joined by enzyme. And it can be joined together in different orders, and which is why it will have different sequence and all. So that after that, the translation will occur at the ribosome. And as different sequence of exons join, there will be different amino acid sequence. And if it has different amino acid sequence, the splicing. It's a splicing thing, okay? RNA splicing. Or how one mRNA can make different proteins, same thing, okay? So the mRNA, it will make the different proteins. And translation will occur, yeah. So as the amino acid sequence is different now, so we know that's what changes the primary structure. So the primary structure will be changed. And polypeptide chains will be produced. So here we have a question. Describe how more than one type of protein. Yeah. So how describe how more than one type of protein can be synthesized from the RNA produced from one gene. Okay, so when a post-translation change or the RNA splicing will occur, and the spliceosome enzyme, it will remove the non-coding introns and it will join the exons in different sequence or in different ways. And that will have different functional mRNA with different arrangement of exons. So that's why it will code for, it will have a different primary structure or different sequence of amino acids. That's why it will code for a different arrangement of amino acids. Okay, or yeah, different sequence or different arrangement of amino acids. And if it has different arrangement of amino acids, a different polypeptide will be formed or a protein will be formed. And then the next one, controlling a gene expression outside the nucleus. So that was for inside, and now this is outside. So there will be further modification of the protein after the protein synthesis occurred. So it can be shortened, it can be lengthened, it can remain intact like it was, okay? And controlling gene expression inside the nucleus, RNA splicing, controlling gene expression outside the nucleus. Okay. So inside, we know the transcription factor. It will bind to the specific sequence of DNA base and have. As you guys have your Edexcel IAL Biology Unit 2 exam in a couple of days, so give your best for the exam and you can join our question paper marathon course for Unit 4, 5, 6, which we have around 2 weeks left. For the upcoming sessions, you can join exam essentials for all units of biology, chemistry, and physics for our high impact crash course for October 25, January 26, or further sessions. Okay, so yeah, it has promoter and enhancer. We already talked about what they are. And promoter, it initiates transcription. Enhancer, it controls the rate of transcription. It enhances transcription. And Yeah, so we are done with controlling inside the cell. Now we will go to RNA splicing. So again, splices of enzyme. It will remove the non-coding introns and join the exons in different sequences, and that will have so versions of mRNA results in different amino acid sequence. And then after inside, it will have the gene expression will be controlled outside the nucleus. So the post-transcriptional, transitional change or the polypeptide change will interact. It can be shortened, it can be lengthened. Sorry, not interact, intact. Why did I say interact? I don't know, but yeah, it will either stay intact or it can be shortened or it can be lengthened, and that will give a variety of protein. Changing genetic material versus change in gene expression. So we have learned both about mutation and epigenetics. So let's go to epigenetics. We did a class on epigenetics, detailed class. So I guess you guys should not have much problem now. So I'll just go through it fast. So methylation and acetylation. So methylation is adding methyl group and acetylation is adding acetyl group. So for methylation, the genes will be switched off. And when it's switched off, we know it's not expressed. And for acetylation, the gene will be switched on and therefore expressed. So for methylation, cysteine, guanine, and crew, histone as well, methylone, lysine. Shown here, we have acetylation, we have histone modification, the lysine, okay. Uncoil, so this can be transcribed now. Different environmental factors. So yeah, different environmental factors, diet, level of exercise, smoking, stress, the affected. And let's go through this question. That uncontrolled cell division, it can result, it can result in the formation of tumor. And the RB gene, it's a tumor suppressor gene. So it will be produced from this gene and to inhibit tumor growth. And the lung cancer can occur. Yeah, lung cancer can occur when this gene has increased methylation compared to blah, blah, blah. So we have to explain why the increased methylation, it can cause the growth of a tumor. So we know methyl group, it's positively charged. It is positively charged when it's added to the cytosine base. And the DNA chain by enzyme DNA methyl transferase, blah, blah, blah. So there will be a change in the arrangement of DNA molecule, and that's why it will physically block the binding of RNA polymerase and the transcription factor, okay? And the silencing gene will be prevented by transcription of gene, and that's why no mRNA will be produced and no RB protein produced, so that's why there will be no inhibition of tumor growth, okay? So it's more like memorizing the points of what is here and what is not. Okay. So cell differentiation, stem cells, stimulus. Okay. So arrive at the cell and acts as a transcription factor. It will bind to a promoter region of the gene, we know stimulus. And DNA acetyl or DNA methyl, okay, differential gene expression. Yeah, and then the transcription factor, it will bind to the promoter region of the switch gene, and the mRNA, it will mRNA transcription will occur, then translation, and then protein will form, and the protein will be permanent. It will permanently modify the cell, or it can determine the function or the structure of the cell. Okay. Now we have different proteins, so yeah, that will change. And how do genes control the phenotype? So we know a particular position on a chromosome where the gene is found. It's called the locus. And the gene linkage, there are two things that we know, gene locus and gene linkage. So it's the linked genes for different characteristics, and it will be found on the same chromosome. Okay, and it will be inherited as if they were a single gene. And there's two types, closely linked and loosely linked. So for closely one, it's transferred during crossing over, And for the loosely linked one, it will not be transferred during crossing over. And swapping a sectional formative, which, yeah, so basically here we see two of them doing crossing over. And after that, they will basically exchange. So it will have the characteristics of the red one and it will have the characteristics of the blue one. So that's how crossing over occurs and all. So for the closed ones, here as this and this was closed, they got changed together. But let's say if we're talking about this gene here and the gene here, they're not close, okay? They're loosely linked. That's why the gene here remains same, but it will be transferred, the other one will be transferred to the other chromosome, okay? And variation, we know there's two types of variation. There is continuous and discontinuous variation. Okay, so for this continuous variation, we know it will not have intermediate, so it will have that specific numbers, or not numbers, more like specific characteristics. And there won't be any intermediates. It could be gender, it could be blood group, it could be number of, which I can't think of anything else. So, and it will be controlled by the genotype. It's represented by bar chart. Okay, so we have bar chart here for male, for female. If it was blood group, it would be A, B, C, O, something like that. Okay, and for continuous, the main difference is that it will have intermediates. So like height, let's say we're talking about height of people. One person can be 5, another person can be 6, one person can be 4.11, one can be 5.11, 4 point something, 5 point something, 6 point something. Okay, there's like intermediate. It's not like exactly 4, 5 or something. There are intermediate, so that's why it's continuous. And it will be controlled by polygenetic genes. Okay, here it was controlled by the genotype, but here it's controlled by polygenetic genes and the environment. Okay, like let's talk about skin color. If we know for people in America or those places, they're fair, but we Bangladeshi people, we have not so fair but not so dark, like a mixture of all. But different people, due to their environment, they have this different skin color. Like in Bangladesh, when it's hot summer, that causes a lot of people to have darker skin and all. So basically lots of environmental factors that causes, and there's polygenetic genes, of course. Like let's say the mother is very, very, very short, the father is very, very, very tall. So the offspring, it might be in between both. So that's how it's polygenetic genes. Or let's say the father has a blue eye, the mother has a green eye, and the daughter that's produced has the intermediate of the eye color. Okay, so that's when it's a polygenetic gene. Weight, same for weight, everything. Okay, so anything that has intermediates, that is continuous one. And studying continuous variation in humans, height, fur, skin color. Okay, one more thing I always tell you, we always need a large sample size. Okay, large sample size is the common thing for both paper, unit 1, unit 2. Okay, large sample size, very, very important. And polygenetic influence by environment. So yeah, of course, polygenetic features are there, but environment, like the diet, energy usage. Let's say people in one country, they eat lots of beef, and people in the other country, they eat lots of, let's say, vegetables or fruits and all. Okay, so their diet will affect their weight and everything, or maybe height or something. So there, and polygenetic inheritance, it's basically when multiple alleles on different loci, yeah, it will determine one characteristics of an organism. Okay, so like there'll be few alleles on different loci, and they will determine a characteristic. Gene for. So here there is 2 questions. So let's go through this. So the sweet pea plants, it was used in an investigation into the inheritance of flower color, pollen grain length, and pea pod shape. So they were controlled by single gene. And sweet peas, they have 7 pairs of chromosomes. So we are given their arrangement. Okay, so we have to comment on the role of meiosis in the inheritance of this traits. Second. Yeah. So meiosis, we know it will increase the genetic variation, of course. So the genes for the pollen grain length, which is here, and the color, the flower color here, they are linked as they're close to each other. We can see they're very, very close to each other, right? So that's why they're linked. And they'll be inherited as one unit. So, okay, whenever they're inherited, the more closer they are, the more chance they have of being inherited together, okay? And the gene for pea pod, we know it's further away and it's not linked. So that's why it will be independent during independent assortment, okay? So inherited during independent assortment, sorry, yeah. And for the other question, the fungus, it lives within journal for a part of its life cycle. The fungus influences the expression of certain genes in the plant cells. Okay, so there was this reduced expression of some genes involved in the DNA synthesis, and there was also reduced expression of genes involved in the synthesis of phospholipids, starch, sucrose. So you have to explain the effect of this fungal infection on the growth of the plant. So we know reduced growth, which is because of the reduced cell division, and because there's reduced cell expression, and that's why there'll be reduced protein production, phospholipids, and all starch, sucrose, all that. And we know their phospholipids, they're used for membrane-bound organelles, and it will be reduced, and the transport of sucrose will be reduced as there's less sucrose. So the less, which I guess my screen's loading later. So yeah, if you see the screen later, then yeah, hopefully that's okay. But yeah, if you have any questions, you can always ask me. And the reduced transport of sucrose, we will know. And if you have less sucrose, there'll be less sucrose transported. And if it's less sucrose transported, it should be available less to the other parts of the plant. And we know it's needed for ATP production. So there'll be less ATP produced, okay? Okay, so this is it for this one. Sure. Elaborate further enhancer. For enhancers, as I told you, to enhance the transcription, so they'll increase the transcription of a gene. And what else do I tell you? Yeah, so they'll bind to transcription factors and they'll regulate the gene expression, I told you, and they will basically increase the rate of gene transcription. Yeah, I guess that's all I can tell you. That's how much you need to know. Yeah, and do any of you have any questions? If you don't have any questions, then I'll move on to the next one. That was quite long. Gene expression. Okay, so I'm moving on to stem cells then. Okay. So stem cells, totipotent, sorry. Totipotent and pluripotent cells. Can someone tell me the difference between pluripotent and totipotent? Anyone difference? Yes, totipotent can differentiate into all types of cell. Okay, which pluripotent cannot, yes. They will only differentiate to embryonic cells and all. Yeah, basically pluripotent, they have this restriction type, they can't differentiate into all types. Very good, yeah. So 1 second. Okay, good. So totipotent cells, so they are the undifferentiated cells, and they have the potential to divide indefinitely, and it will develop into all specialized cells. Okay, they have no limit, but for pluripotent, they have a limit, and happens after cleavage. All cells are. So this is about the stages. So basically, morula, embryo, empty ball of cell. And here, from here, some part of it will divide to become the placenta, and all the cells in the morula stage, they're totipotent, okay? And then that will become a blastocyst. So the outer layer of cell, it's called the trophoblast, which will form the placenta. And these cells here, they're these cells here, they're pluripotent cells, okay? And the source of embryonic stem cells, yeah. So all these cells in the blastocyst, these are pluripotent cells, okay? And for morula, they're totipotent cells, okay? And all cells are pluripotent. So as already Rakshi told, that it's an unspecialized cell, and they can divide indefinitely, and they can give rise to many specialized cells, but not all, okay? So as Rakshit mentioned about the extra embryonic one, it cannot give rise to extra embryonic one. So it basically tells us that it has a limit to the number of or the type of cells it can divide. Acha, cleavage, acha, Rakshit, acha, yeah. So basically cleavage, if you ask me what this is, cleavage is like, you know how the cells, they're dividing after, yeah, the cell division, this dividing thing after fertilization, and they're forming, it goes from a zygote to an embryo, okay? The division and the process of going from a zygote to an embryo, that's called the, what's that called, cleavage thing, okay? And describe the structure of blastocyst. So this time, sorry, not this time, we already did this question. So the structure of blastocyst, we know it's the outer layer. So it has the outer layer, which is called the trophoblast. And it has an inner mass or the embryoblast, and that contains pluripotent cells in the inside. And they basically have a hollow cavity inside, which is called the blastocell. Here, this hollow cavity, they did not, it's not labeled here, but this hollow cavity, this is the blastocell, the inner mass, or yeah, the embryoblast or inner cell mass. It will have pluripotent cells, and the trophoblast, the outer layer. Sure, trophoblast cells are totipotent. Not exactly. They're not totipotent, okay? Trophoblast, you cannot say they're totipotent, okay? And then types of stem cells. So there are embryonic stem cells, there's umbilical stem cells, and there's adult stem cells. I think I did a class on this one also for stem cells. So you guys should not have much of a problem. So I'll just go through it. So I told you embryonic stem cells, they're totipotent. cells, and then umbilical ones, umbilical stem cells, they're stored by freezing. And yeah, so it's between the placenta and the fetus, so they will have pluripotent cell. So they take up a lot of space. Pension can cause cancer. Okay. And adult stem cells, they're basically found in small number of different cells in tissue organs, multipotent. So yeah, also adult stem cells, they're multipotent. Okay, so they have limited specialization and they're hard to extract and hard to find to excuse for specific specialized cells so in lab. Okay, so yeah, basically they're hard to find and all or hard to extract. Okay, stem cell therapy. We know there's cloning, there's IPS. So for cloning, it's just copying or cloning the word that we know. So it can be cloned into kidney cells, spinal cord, embryo, blah, blah, blah. And for IPS, it basically goes from totipotent to pluripotent to adult stem cells, okay? And iPS cells are similar to human embryonic cells. And we get this by genetic engineering from adult to pluripotent. And hard to control gene, okay? Yeah, so we know cloning, it can cause cancer. This is important. So it can cause cancer and it is hard to control the gene. For it's different. And the advantage you could say is that there's this rejection in the organ transplant. So if you do cloning, there's a rejection thingy. So that's a good thing. For IPS, it's hard for IPS to differentiate cells tendency to be cancerous. Again, both of them, they can be cancerous. And we know for the ethical objections that people have. For IPS, there's no ethical objections basically. And again, there's no rejection, okay? So that's what advantage for both. So just why is it difficult for a specialized cell to become stem cells, okay? So gene, we know it's switched off permanently. And yeah, so gene, when it's switched off permanently. And the cell, it will be permanently modified. And yeah. So regulatory authorities for embryo research. Actually, you need to know this so that we need to monitor the research. And we have to issue license for stem cells, make sure they use early embryonic stem cells and prevent unethical use of stem cells. Make sure stem cell source is ethical. Same thing, the last one is same. So yeah, this is it for this part. I didn't include much here because we already discussed in details about stem cells in one of the class. So if you guys need more clarification, you can tell me and I'll give you guys a recording for that class so that you guys can go through it. And yeah. So do you guys have any questions? If not, I will move on to the next one. So mitosis and meiosis, I will go on to mitosis and meiosis. Okay, what do we have here? Mitosis, meiosis. I'm sure you guys know already a lot of it, but I'll just go through what's important so that you guys can have a recap. So we know the structure of chromosome. This, yeah, this is one chromatid, this is one chromatid, and they're joined by a centromere. And two chromatids together, they are called chromosome. And they're made from two sister chromatids that is attached to a centromere. And it's made of two DNA molecules. You know this. So interphase, chromatin. So yeah, the uncondensed, unfolded DNA. And it will basically just replicate, semi-conservative replication will occur. And two DNA molecules will be produced. And we know for each chromosome that is made, for each chromosome that is made, two identical structure called chromatids. Two chromatids make one chromosome, and it's formed of a DNA molecule that's associated with the protein called histone, and it will be held together at the centromere. And chromatin, it's a mixture of DNA and protein. Okay, so chromatin that we're talking about, chromatin, and this ultracell structure, I told you it's inside the nucleus, chromatin. So yes, that is a chromatin. Function of cell cycle. Actually, let's go to this one. Okay, so we are seeing the chromatin in phases of the cycle. So we see the double chain of deoxyribose here. This is the deoxy DNA chain or DNA strand. And the chromatin. The second picture is the chromatin with the histone proteins, the circular ones. It's the histone proteins. And the third one. Yeah, so third one, it's a chromatin interphase. And the fourth one is the dense chromatin during prophase. Because we know in interphase that it will double up, so yeah, or it will replicate. And then number 5, it's the chromosome at metaphase, okay? So here they're talking about the differences. So chromatin and chromosome, they are the same, but yeah. Chromatin is the unfolded, uncondensed DNA. But when the cell division occurs, we can see chromosomes, which are the folded, condensed DNA. And we know chromatin is found through interphase, and chromosome is found during mitosis. Okay, the one that saying when during cell division, the chromosome it becomes shortened and also yeah, it becomes condensed and shortened and fatter. Okay. And cell cycle. Let's go through the cell cycle. We don't have much time. Which chromosome? Yeah, it is, okay? It's made of two identical double helixes, okay? DNA double helix, yeah. Cell cycle. Is the DNA included now? Yeah, it's mostly chromatin, but only during cell division it becomes a chromosome. Okay, and then again after it's done, it becomes a chromatin. Okay. Okay. So we can see the cell cycle. We know interphase, it has 3 phases, G1, S, and G2. So the whole thing, the bigger phase is the interphase one. Then there's the mitosis part. And a very small space is taken by cytokinesis, where the cytoplasm divides, okay? Cytokinesis is the division of cytoplasm. Karyokinesis is the division of nucleus. Okay, and we know in the S phase, the DNA replication occurs. So enlarged cytoplasm and cell continues to grow, and new DNA is checked for error and repaired. Yeah, so the DNA is also checked for error and repair. Okay, like if there's error, it's repaired, and the DNA will replicate, will double. The number of chromosomes does not change. Okay, and nuclear division mitosis, this. So in interphase, organelles, they are being copied. Protein synthesis occurs. We know increase in cytoplasm. And number half to produce energy for growth and okay. So yeah, mitochondria, it's also replicated during interphase so that the number of mitochondria can be, when the cell division occurs, it can be half later and it can still provide enough energy for growth and mitosis, okay? So functions of cell cycle, we know there is a sexual reproduction, replication of DNA, increase in cell size, growth, replacing dead cells, replacing damaged tissue. So role of cell cycle in producing new cell. And when it synthesizes new organelles, increases the cell size, in replication of DNA, all that. Okay, so now we're talking about we're getting the depth of the stages of mitosis. So first one, we know it's there's prophase, there's metaphase, there's anaphase, there's telophase. Okay, PMAT, which basically means PMAT. If you want to remember it like that, PMAT. So for prophase, it's again divided into two parts. Okay, early prophase, late prophase, or if you want, you can just say prophase as prophase, and then basically four parts: prophase, metaphase, anaphase, telophase. But prophase is again divided into two parts. The early prophase, we know the centrioles. Yeah, so the centrioles are replicated. The chromatin, it will condense into chromosomes here. Remember, it was as Rakshit asked, it was usually chromatin, but then during cell division, it will become into chromosome, and they'll become visible and shorter and thicker. Okay, the one they're saying shorter and fatter, or yeah, shorter and thicker. And the nucleolus and the nuclear membrane, they will all, nucleolus and the nuclear membrane, they will all disappear or break down or whatever. And in the late prophase, so after the centrioles were replicated in the early prophase, the centrioles in the late prophase will move to opposite poles of the cell. Okay, centrioles move to opposite poles of the cell in late prophase, and the nucleolus, nuclear envelope disappearing, same. And the chromatin, it will condense into chromosome. They will become shorter, thicker, visible. Nucleolus, this. Yeah, so the centrioles that move to opposite poles, they will start forming spindle fibers, okay? So they'll start forming spindle fibers. And then in metaphase, should we have 4 minutes? Okay. In metaphase, the centrioles, it will reach the pole of the cell. Yeah, here, once let me, this centrioles, it will reach the pole of the cell, and the spindle fiber will be formed, or it's formed. The chromosomes, here they will line up in the equator, and the spindle fiber that's formed, it will attach to the chromosomes in their centromere, okay, in the middle part, the centromere, it will get attached to there. Okay, so that's metaphase. For anaphase, the spindle fibers that was attached, now they will start to pull, they will shrink and shorten, and when that happens, they will start to pull the chromosomes to opposite poles, okay? So that will break the centromere, and the sister chromatids, they will be pulled apart. So there's two sister chromatids you can see, and as they're joined by centromere, it will break. Now there will be no joining. So one sister chromatid will move to this side, one sister chromatid will move to the other side, okay? And that will be pulled by the spindle fiber, which is becoming shorter and it's shrinking. So yeah, it will just pull them to opposite poles. And yeah, that's what metaphase is. For telophase, now that they're being pulled, now they're done being pulled, the spindle fibers, their work is done, so they will be broken. The nucleolus and the nuclear membrane that broke down in prophase, that will again reform, and the chromatids, it will uncondense and it will form chromatin. Okay, so the prophase and the telophase, they're kind of like opposite. So whatever got broken down, whatever changed into what, will be returned back to their normal stage. Okay, so at prophase, the nucleolus and the nuclear membrane, they broke. Now it will reform in telophase. The chromatin, they form chromosomes. Now the chromatids that are formed, that will be changed into chromatin. Okay. For cytokinesis, the last one, the one that's saying division of cytoplasm. So that like cytoplasm will divide, and there'll be two different, or not different, basically genetically identical cells. Okay, there'll be two different cells formed, but they'll be genetically identical to each other, and they'll have the same number of chromatin. Okay, because when it was pulled, they were broken apart and pulled to opposite directions, so it will have the same number of chromatin. In animals, cleavage forms. In plant, cell plate forms. Okay, so that's how we differentiate the cleavage and the cell plate. Can we say centrosomes instead of centrioles? Actually, centriole and centrosome. Yeah, you can say centrosome, because centrosome is basically when two centrioles are joined at right angles, the one that is saying. So that's centrosome. And individually, they're called centrioles. Okay. And we have interphase, mitosis, cytokinesis. Okay, so interphase, we know this chromatin. It will have one DNA molecule, and in prophase till metaphase, there'll be two sister chromatids. So as I say, each of them will have one. So there will be a total of two DNA molecules. So we're done till this. So meiosis, we know meiosis, it will result in haploid nucleus. It's the same as mitosis, but for meiosis, we have all of the process repeating twice. Okay, like prophase 1, prophase 2, like prophase 1, metaphase 1, PMA. So anaphase 1, telophase 1. Again, the prophase 2, metaphase 2, anaphase 2, and telophase 2 will occur. Okay, so the second ones are the same, but the first one, there's a slight difference here. So there'll be two divisions, and the genetically different. Four cells will cross over, and then there'll be independent assortment. Okay, so crossing over, independent assortment, that's what creates the genetic variation, and that will occur in meiosis. Okay, and it will result in haploid nucleus, and yeah, and as they have haploid nucleus, n number of nucleus, so n plus n will give us 2n. So that's why n and n can fertilize together and form a constant chromosome number. So yeah, the prophase 1 and metaphase 1, let's see what happens to create the genetic variation. So basically the homologous pair of chromosomes, they will pair up. So it's going to be like this. One chromosome, two chromosome, another, let's see, my chromosomes don't look like chromosomes, but yeah, these are chromosomes. Okay, so there'll be one pair of homologous chromosome, and yeah, pairing of homologous chromosomes, and they'll be crossing over, and where the wrapping section of chromatids, the recombinant parts of which chromatids are the chiasmata. So the one that I showed you, the crossing over part, so here, this, and this one will occur, crossing over will happen here. So yeah. And the metaphase one, the homologous pair of chromosomes, it's like the usual one, they will line up in the equator. and then independent assortment will occur, and the chromosomes will arrange themselves randomly. And yeah, as this random arrangement, there'll be new allele combinations, so that will produce genetic variation. Okay. This to crossing over and independent assortment. So crossing over in prophase 1, independent assortment in metaphase 1. Okay. And that's it about mitosis and meiosis. So let's discuss plant gamete formation. Okay, so we know the gametogenesis will form two things, the microgametogenesis and megagametogenesis. And micro is for the pollen grain, mega is for the egg cell. So for meiosis 1, one cell dividing into 2, meiosis 1. And that individual cells will divide into more of them. So that's meiosis 2. So 4 daughter cells. And for megagametogenesis, the same thing basically and all. So 1 will divide into 2 and 2 will divide into 4. And so at this pollen, they will divide by mitosis. So the pollen tube nucleus, generative nucleus. So yeah, we know there's two nucleus, pollen tube nucleus and generative nucleus. And yeah, so the two male gametes. And for mitosis. Yeah, so mitosis will occur and will divide. So there were 3 mitotic divisions. So from 1 to 2, 2 to 4, 4 to what's it? 1, 2, 3, 4. Yeah. Okay, so there'll be 2 polar nuclei. This is the female gamete, this is the embryo sac, and this is the ovum basically. So the pollen and the ovum, here we can see the pollen tube nucleus, the end, the 2 generative nuclei, and it spikes to what? Stigma. Actually, yeah, it will stick to these spikes. The covering or the membrane has spikes, and that will help it to stick to stigma. And for the ovum, it will have 2 polar nuclei, and it will fuse into, fuse with the generative nuclei. It will form the triploid endosperm, and the female gamete will fuse, and the male gamete with the male, actually, yeah, the female gamete, male gamete, we know it fuses, and that will form the diploid zygote. Okay, so that's how it's 3. And we have more. So nothing much for plant gamete formation. So, Acha, if you guys have any questions, you can write it. Till then, Rakshit wrote, what is the role of the 3 synergids in the ovary? Basically, the 3 synergids in the ovary, they help in the fertilization mostly. Okay, with the pollen tube and the fertilization thingy, they're involved in that. And they will just, how do I say, it will move the pollen tube to the egg cell or the ovum, and yeah, and after the fertilization, it will break down, not exactly break down, yeah, it will degenerate. Okay, so yeah, after fertilization, it will degenerate. Okay, that's all I guess you need to know. So yeah, the pollen tube thing is the fertilization, and then after it's done with fertilization, it will degenerate. Okay, so it will create a path. It will move the pollen tube to the egg cell, and yeah. And we know when it goes to the egg cell, there will be fertilization. So that's how it helps in the fertilization. And then, yeah, after that, they're done. Okay, they're done with their role, so they will become degenerated. Okay, and that's all for plant gamete formation. Do you guys have any questions? Or else I'll go to the human gamete formation. Oopsie. Yeah. Oh, okay. As you guys don't have any questions, I'll. Wow. Okay. Yeah. So I'll start with the human gamete formation then. So for this one, again, the gametogenesis. So gametes, we know they're the sex cells and they have half the number of chromosomes, so that when they fuse together, the half chromosomes, they will make a complete set of chromosomes. And yeah. So gametogenesis, formation of gametes. It's basically gamete and genesis means formation or making or production. So yeah, making of gametes, formation of gametes, yeah. And it will form oogenesis and spermatogenesis. So oogenesis, eggs, ova, spermatogenesis, sperm. So the mitosis. And yes, they'll form four here. And only one of them will survive and the rest three will die. But for sperm, there's four. And yeah, so the polar bodies that we know, they will die. And only the bigger one, it stays. And axon, fat few fixed. Okay, what do we have? So primary spermatocyte, secondary spermatocyte, and you'll form four spermatids. And from that, it will become spermatozoa. So four spermatids will form four spermatozoas. And yeah, so one of them, as I say, one of the three polar bodies will die, one of them will leave, and it will again divide. So cell membrane, zona pellucida, we have learned about zona pellucida a lot. So that's important, zona pellucida part. So it's that covering, and this follicle cell, cortical granules, haploid nucleus, because it's a gamete, and cytoplasm. It will contain lipid as energy storage for fetus, because fetus needs energy. So the lipids that stored, it will be used as the energy storage. And for the sperm cell, for this one, the egg cells, they're fat, they're few in number, and they're fixed. For sperm cells, they're many in number, they're mini, and they're motile, okay? Because they have a tail, so that's why they're motile. But egg cells, we know they cannot move, so they're fixed. And this is what a sperm cell looks like. They have acrosome at the head, and it contains acrosine. Yeah, it contains acrosine. And this part, the middle part, has mitochondria, and that basically supply energy to the tails, so that the tail, it is swimming to the ovum, right? Like it's motile, so it can swim to the ovum, and that's why it needs energy. So that's where the energy comes from. And acrosome in the head contains acrosine, and it will digest the zona pellucida and it will fertilize the cell. Okay, so basically it will come here, digest the zona pellucida, and it will go inside. Okay, so that's how it enters. So the tail-like thing is the flagella, and that provides the motility. So the flagella is what helps them move. And acrosome and lysosome. So the similarities of acrosome and lysosome is that both have hydrolytic enzymes or digestive enzymes. We already talked about lysosomes, and now we are talking about acrosomes. So both will have hydrolytic enzymes. and both are single membrane bound organelle. Okay, so as both are single membrane bound organelle, both will have single membrane basically. And the difference is that we know lysosome, it was the circular one, and acrosome, it's not of course circular. For lysosome, it was involved in the intracellular digestion, and acrosome, it's for the extracellular digestion. And a lysosome, we know it breaks down the cell components, and the acrosome, it basically digests the outer layer of the egg, which is the zona pellucida. Okay, so the role is kind of different, and one works internally, one works outside, and one has a spherical shape, one does not have any. Like it's not spherical, so it has curved, or it does not have a, you can say it's not spherical. So yeah, do you guys have any questions from here? Human gamete formation. If not, then I'll move into fertilization. Okay, this part we already did in the class. Well, actually, let me still go because we did a lot about fertilization and all. So fertilization, we know it makes the full complement of chromosome number. And we know it needs to be restored. It will produce genetic variation, fertilization, because two cells are fusing. So both the characteristics are fusing. So yeah, it has variation. And we know why it has variation. The meiosis part thing also. And without fertilization, the number of chromosomes wouldn't be constant, because we see in the gametes, they have half the chromosome number. So two gametes, half and half, they fuse together and they make a full complete set of chromosomes. And like, or else the number of chromosomes would have been doubling every time. So if like N and N, like two complete sets, would join together to form 2N, then more 2 would join together and then it would keep on increasing, okay? So just to keep it keeps it constant, number of chromosomes and all. And we have two ways, one is in humans, one is in plants. So let's go to the human one. So we know the sperm, with its tail, it will swim to the not the nucleus. It will swim to the egg cell and it will digest the. Okay, so the sperm, it touches the eggs and triggers, okay, reaction. So yeah, the acrosome, there will be reaction and all. So it will release the acrosome, and it will, like, the acrosome will release the acrosome, and we know that will digest the zona pellucida. So here you can see they're trying to digest the zona pellucida. Yeah, here. And then after it's digesting, it will penetrate inside the egg cell, and that's how the fusion occurs. So there will be a cortical reaction. So the cortical granules, it will be released by exocytosis, and that will combine with the zona pellucida, and it will harden, okay? Yes, it will harden so that more sperm cells, they don't fuse with the egg and polyspermy does not occur, okay? So zona pellucida is digested. So the fertilization membrane, it's formed by reaction of cortical granules and zona pellucida. So it's hardened and all. And yeah, so that other sperm cells, they cannot enter. And chromosomes they're about to undergo division by mitosis. So yeah. So that's for the human one. For plants, yeah, we know for plants, the pollen grain, it will land on the stigma, and the pollen tube, pollen tube nuclei, it will start to release enzymes, and it will digest the style's tissue, and there will be a pollen tube that will go through the style, okay? So the tube that's created, it will go through the style, and that makes a pathway for the male gamete to go to the embryo sac, okay? Yes, Simra?
I need to leave right now.
Oh, you have to? Acha, do you have any questions from Acha, we'll finish fertilization and we'll be done. So yeah, if you have any questions, you can tell them, or else, yeah, we'll just do this part and we'll be done for today, okay? So yeah,
Okay.
if you want, if you have to leave, you can, okay? Also, we're just done in like what, 2-3 minutes. If you can't stay, then that will be good. But yeah, if you have to, then okay.
Yeah, okay, let's do it.
Yeah. So the importance is that, yeah, it will make a pathway so that the male gamete can go to the female gamete and fuse. And yeah, and it opens the micropyle. So this is the pollen tube here, and it will go here, and it will come inside the ovum, and yeah, it will fertilize. And the male gametes, it enters through the micropyle, and here, this thingy, the outside covering thingy, it's the integument. And it surrounds two placenta. Sorry, yeah, it surrounds the placenta. What am I saying two? Yeah, it surrounds the placenta, okay? And the double fertilization. So the male gamete, it will fertilize with the female gamete and it will form a diploid zygote like we know. And then another second male gamete, it will fertilize the two polar bodies. And as it fertilizes two polar bodies, and one male gamete, so it will have triploid endosperm. Okay, and that basically provides the food storage or energy, okay, for the so that it can grow properly. So here, do we have anything? Yeah, so the pollen tube nucleus, it will degenerate after the tube enters the ovule, and the first male gamete, we already told you, it will fuse with the female gamete nucleus and it will form a diploid zygote. And the second male gamete, it will fuse with the two polar bodies or two polar nuclei, and that will form an endosperm, triploid and primary endosperm nucleus. So that's going to be 3N, okay? And advantages of self-fertilization, disadvantages. This one, we already did this question in the class. So do any of you have any questions from the advantages, disadvantages? Actually, let me just go through it if you guys want. So we have 6 minutes left. So yeah, I can go through it. So advantages is that we know for self-fertilization, there'll be, sorry, if we prevent self-fertilization, mostly it's, if self-fertilization does not occur, then there'll be genetic variation because it's not, the cell is coming from the other plant or something. So yeah, that's it. It will have genetic variation. And we know the inbreeding depression can occur for self-fertilization, so that will be prevented or the risk will reduce. And increased genetic variation, we know if there's a change in the environment or there's a new selection pressure, let's say suddenly the environment becomes hotter or tall trees are growing or whatever, some environmental changes or selection pressure, if it occurs and if it has variation, so there will be chances that the selective one, it's already there in the population. So the advantageous allele, it's there in the population, so there will be more chance for those to survive, and they will adapt to the new environment or the selection pressure, and they can leave. Okay, or else if there was no genetic variation, if one disease or one type of selection pressure acted on them, and that plant or that certain thing does not have the advantage, then all of them would have died. Okay, so that's one thing. And the chemical releasing plants, it was in the question that they produce both ovule and we know that the ones that cannot self-fertilize, sorry, the ones that self-fertilize, they have both ovules and pollen. And the disadvantages of preventing self-fertilization. So that was the advantage, and disadvantage is that there'll be reduced chance of pollination or fertilization because let's say one plant, it's not found a lot. Like let's say 1 million, one plant, it's very rare, so it's not really found, okay? And if it cannot self-fertilize, it has to look for another plant of the same species. And if it's very far away, we know by wind pollination or insect pollination or whatever it is, it's going to be really hard for that to reproduce more, right? So that's why it reduces chance of pollination or fertilization. And there has to be both the male and the female date plants in the habitat. The one I was saying that both has to be there if it's farther away, or let's say There's no other plant, so that will reduce fertilization and consideration of lack of insect pollinators. So yeah, if there's less insect pollinators, how are they gonna go right from one plant to other plant and fertilize? So yeah, and successful pollination does not occur. So there will be no seed production, and the plant will become endangered, and slowly it will become extinct and all. And the offspring may not inherit yeah, the one that's saying that if it does not have the advantageous allele, so the mutation can cause it to all of them to die together. Okay, so they'll be extinct. And advantages of self-pollination is that we know fertilization will occur because both the male and the female gametes, they're in the same plant. So that's where the fertilization will occur definitely. And the reduced risk of extinction. So yeah, as fertilization will occur, there's no risk of extinction. And the genetically different ones. Actually, this was in the question that Danelian cells are genetically different. So that's why the seeds will be different also. And even though, yeah, and for disadvantages, there will be no genetic variation. That's the usual one. We know that if the gametes are from the same plant, they will have the characteristics of the same plant too. Okay, there will be no variation, and that's why it can reduce the genetic variation, and it will be, if there's a new selection pressure change in environment, they cannot survive. And yeah, so that is it. Inbreeding. Rakshit, inbreeding is basically when two, how do I say, two genetically identical or like similar organisms, they breed together, okay? That's inbreeding, okay? When two of them, similar ones, they breed together, that's called inbreeding. That's like breeding what? Siblings and all, maybe? I guess that's how you can say. Like very close individuals, okay? Yeah, breeding two close individuals. Okay, guys, we have 1 minute left for the class. So if you guys have any questions, you can tell. All right, that will be it for today's class. Simra, if you have any questions, Simra is here, right? Yeah, if you have any questions, you can ask, or else you can leave. That's okay. And others, same to you. If you guys have any questions, you can ask, or else we'll discuss the topic for in the next class. Okay? Any questions? Any questions? Any questions? And I already gave you the notes. So yeah, guys, please try to review these. And as I already told you for unit 1, that when I'm discussing this, I give it to you before. I give you the notes before so that you guys can mark the new parts that you're looking at and you can review them later because new parts are harder to remember. Okay, the ones that you know, that's great. But if you don't know something, mark them, and after the class,